So how many students we shall have?
Close to 350, sir. Some are joining from classrooms and the rest are joining from their personal mobiles.
Okay, okay, fine. So how many faculty you have in biotechnology?
33.
27 members, sir.
Okay.
27.
How many professors, how many associates, and how many assistants?
Professors, 6 members, sir. Associate, 13 members, sir. Remaining all assistant professors.
Okay, okay, good. Very good. So it is 11. Shall we start?
Okay, sir.
Or shall we wait for someone?
No need, sir. Indra madam will
No need.
introduce you, sir. Indra madam, please.
Okay, fine. Go ahead.
Good morning to one and all. I am Dr. M. Indira, Associate Professor in Department of Biotechnology. It's a great privilege for me to introduce today's speaker, Dr. P. Anand Kumar. Dr. P. Anand Kumar is currently working as Scientist Emeritus at Indian Institute of Rice Research, Hyderabad. Earlier, he served as Director, National Research Center on Plant Biotechnology, New Delhi. He obtained MSc degree in botany from Sri Venkateswara University, Tirupati. He joined as a scientist in the Agriculture Research Service of Indian Council of Agriculture Research in the year 1978. He obtained PhD in plant physiology from Indian Agriculture Research Institute, New Delhi. After working as Alexander von Humboldt Fellow in the University of Hanover during 1991 to 1992, he moved to Biotechnology Center, IARI, in the year 1993. He specialized in the area of transgenic development for insect resistance, utilizing the genes encoding insecticidal proteins of Bacillus thuringiensis. Dr. Kumar developed fruit borer resistant brinjal and tomato, which were licensed to private companies. He was also involved in the development of pod borer resistant pigeon pea and chickpea. Dr. Kumar published over 150 research articles, books, and book chapters. He has obtained 3 patents on codon modified and chimeric Bt genes. His current research interests include functional genomics of aerobic rice. Dr. Kumar serves as an editor, GM Crops and Food. He was secretary to Society for Plant Biochemistry and Biotechnology, India. Some of the honors and awards received by Dr. Kumar include Young Scientist Award by Indian National Science Academy in the year 1989, Fellow Alexander von Humboldt Stiftung, Germany in the year 1991, Professor Hira Lal Chakravarti Award by Indian Science Congress Association in the year 1996, Fellow National Academy of Agriculture Sciences in the year 1997, Vasvik Award for Agriculture and Industry in the year 2000, National Bioscience Award by Department of Biotechnology in the year 2001, Recognition Award by National Academy of Agriculture Sciences in the year 2005, Sardar Patel Best ICAR Institute Award in the year 2010 as Director of NRCPP, Fellow National Academy of Sciences, India in the year 2011, Dodla Raghav Reddy Medal by Plant Protection Association of India year 2014. So thank you, sir, for accepting our invitation. Now I request Dr. Anand Kumar to give his talk on what is Bt Brinjal, why do we need it? Sir, please.
Thank you, Dr. Indira Garu, for your nice, generous introduction. I also take this opportunity to thank Professor Kavi Kishore, who has been a long time friend of mine, more than 35 years or so. So we start the talk with the title that is, what is Bt Brinjal and why do we need it? As you all know, India is the second largest producer of fruits and vegetables after China. And there is a tremendous diversity in vegetable crops in India, as you can see from this picture. And most of the vegetable farmers in India are marginal and resource poor farmers who depend on vegetable selling for day-to-day income and also for modest savings. And as you know, as in other crops, vegetable crops also face lot of biotic stresses, starting from weeds up to mycoplasma diseases, out of which the insect pests are the major constraint or the stress factor, destabilizing the yields of vegetables and fruits. And in today's context, when we talk about brinjal, as you all know, brinjal shoot and fruit borer, also known as Leucinodis orbonalis, is a serious pest on eggplant or brinjal. As you can see, the larval stage of shoot and fruit borer bores into the fruits and make them unmarketable and also unpalatable. So much so, in India, we use a tremendous amount of pesticides in vegetables for insect pest management. As you can see, brinjal receives the most amount of pesticides after chilies, followed by okra. And for your information, on an average, a brinjal farmer in India sprays pesticides 40 to 50 times in a single crop season. So much so, the pesticide residues in vegetables is largest in brinjal after cabbage and cauliflower. And you know very well, most of these pesticides and pesticide residues are harmful to human health and causes many diseases, including cancer. So we have to practice insect pest management in an eco-friendly and sustainable manner. So for that, we have to depend upon nature to use natural or biological insecticidal proteins. So there is a long list of insecticidal proteins found in nature in many organisms, out of which the delta endotoxins of Bacillus thuringiensis assume tremendous importance and significance. So what is Bacillus thuringiensis? It is also known as Bt in short form. Bacillus thuringiensis is an ubiquitous soil-dwelling, gram-positive, sporulating bacterium. So if you see the bacterium in a scanning electron microscope, you see a large spore on one side and a parasporal body, light blue in color, in the bacterium. This parasporal body, also known as crystal, is nothing but a tightly packed structure with millions and millions of bipyramidal diamond-shaped crystals. And what are these crystals? These crystals, in turn, are tightly packed protein molecules. You can see the three-dimensional structure of this protein molecule, which is delta endotoxin. So how this delta endotoxin or insecticidal crystal protein of Bt works on agriculturally important harmful insects? You see here, for example, a typical Lepidopteran larva that is consuming a leaf on which you have Bacillus thuringiensis as well as crystals. The crystals, they go inside the midgut of the larva, which is highly alkaline. The pH can range from 10 to 12. And these crystals are solubilized into individual delta endotoxins. And you see these delta endotoxins from this cartoon, you can see a red part and a black part. The black part is the C-terminus half of the protein. It is digested away by insect proteases in the gut. Whereas the red part, that is the N-terminus part, immediately goes and binds to the insect cell membrane receptors, usually aminopeptidases. So once the toxin, the N-terminus half of the toxin, goes and binds to the receptor in the insect cell membrane, instantly a perforation or a hole is formed in the cell membrane. So once the hole is formed, it becomes permeable to ions, sugars, and small molecules, so much so the cell swells like a balloon and simply bursts. So millions and millions of such pores are formed inside the gut of the larva, and within one day, the larva dies. So most of the agriculturally important pests belonging to orders Lepidoptera, Diptera, Coleoptera, fortunately for us, are highly susceptible to insecticidal crystal proteins. And I would like to tell you at this juncture that this particular receptor to Bt is not present in human beings, other mammals, other vertebrates, invertebrates, and soil flora, fauna, beneficial insects, and non-target insects. That is what makes Bt totally safe, totally harmless, innocuous to human beings and other organisms, at the same time highly insecticidal to agriculturally important pests. So, if you see the genetically engineered crops, among which the insect pest resistant crops occupy an important part. For example, in 2019, GE crops have been cultivated in 191 million hectares in 29 countries, and 44 countries imported the GE crops or food products or food produce thereof. The major crops are soybean, maize, cotton, and canola. And the major traits are herbicide tolerance, followed by Bt crops or insect pest resistant crops. So, if you see the data in 2019, global genetically engineered food crops are about 11, starting from maize, going to eggplant, soybean, canola, sugar beet, papaya, squash, potato, apple, sugarcane, pineapple, and ultimately brinjal. These are the genetically engineered food crops currently cultivated in 29 countries of the world. Eggplant is cultivated in Bangladesh. So much so, if you see the data from 1996 to 2016, it shows that the GE crops have resulted in tremendous economic and environmental benefits to humankind. Of course, I'm not going to read all these things since we don't have much time. So I go to the next slide, but I'm going to give this PowerPoint presentation to the department and you can all share it among yourselves. So much so, in 1995, we have developed in our laboratory a transgenic brinjal or genetically engineered brinjal using an insecticidal crystal protein gene by name CRY1AB. And these plants have been developed and tested in glass houses. And also concomitantly, we have done all molecular analysis, insect protein estimation analysis, and also insect bioassays. And we have also done a small field trial in the farm of Indian Agricultural Research Institute away from the plant breeding plots. And the plot has been covered with 2 layers of mosquito net that facilitated introduction of brinjal shoot and fruit borer moths inside so that you can assess the efficacy of the insecticidal crystal protein expression. We found mixed results. In 1 year, we got good results. In the second year, we did not get very good results. Subsequently, in the year 2002, a private seed company by name Mahiko, located in Jalna, Maharashtra, has used an insecticidal crystal protein gene encoding CRY1AC and developed Bt Brinjal hybrids that are shown here. These hybrids have been subjected to all kinds of biosafety tests. As you can see here, the transformation of brinjal has been done in the year 2000, greenhouse evaluation in 2000 and 2001, molecular characterization, followed by biosafety testing, field testing, large-scale field testing, And ultimately, after ascertaining the safety, biosafety and environmental safety of event EE1 of Bt Brinjal, the Genetic Engineering Approval Committee located in the Ministry of Environment and Forests, New Delhi, has approved it on October 14, 2009. However, in October 2009, the Minister of Environment and Forests, by name Mr. Jairam Ramesh, announced public consultation. After public consultations, he imposed a moratorium on Bt Brinjal event EE1 for a period of 10 years. Meanwhile, what happened? The Mahiko company has provided event EE1 Bt Brinjal to Bangladesh, mediated by Agricultural Biotechnology Support Project of Cornell University. The government of Bangladesh has seen the data, the field trial data of Bangladesh Agricultural Research Institute done for 2 to 3 years. And in the year 2014, the government of Bangladesh approved 4 varieties of Bt Brinjal for cultivation by the farmers. So much so, in the past 5 to 6 years, the brinjal farmers of Bangladesh have enjoyed tremendous benefits, social, economic, and environmental benefits in Bangladesh. So, simultaneously, what we have done in the year 2000, we have identified an insecticidal crystal protein by name Cry1FA1. It is present in Bt strain Isawai, and we found that this particular Bt toxin is highly effective against brinjal shoot and fruit borer, diamondback moth, a pest on cabbage and cauliflower, maize stalk borers, tobacco caterpillar, pink bollworm on cotton, cabbage loper on cabbage, and cotton bollworm on cotton, and spotted bollworm on cotton. And we have constructed a codon modified Cry1FA1 suitable for expression in plant cells, high expression in plant cells. And this codon modified synthetic version of Cry1FA1 gene has been mobilized into brinjal, an IARI variety, 45 years old variety by name Posa Purple Long, a variety which is very popular even today. And the codon modification has been done so much so the AT by GC content is shown here. AT content is 54%, CG content is 46%. And we have taken a patent granted by India Patent Office in the year 2010 on this codon modified version. And we have mobilized this in Bt Brinjal, and we have identified lines that have single transgene insertion. We have also analyzed the flanking sequences on both sides of the transgene insertion. Later, for the purpose of plant breeders, we have also developed homozygosity primers, and then this event 142 has been licensed by IARI to Bij Shetal, a vegetable seed company located in Jalna in the year 2005 on public-private partnership basis. This is, as you can see on right-hand side bottom corner, it is totally a Make in India product. And the company has subjected the event 142, as you have seen earlier in the Mahiko's Bt biosafety slide, all kinds of Bt biosafety, environmental safety, and other safety parameters. The company has done this over a period of 5 to 6 years. So ultimately, they have done biosafety research level 1 trial at 3 locations: Jalna in Maharashtra, Benares in UP, and Guntur in Andhra Pradesh. This BRL1 trial has been done for 2 consecutive years. This particular data shows, you see the last column, shows the mean % damage of brinjal shoot and fruit borer in Janak Bt, 0.82%, BSS 79 Bt, 1.39%. In contrast, the normal control Janak non-BT 26% damage. Similarly, non-GM control BSS 793 non-BT 24%, and a local non-BT Brinjal check had 21% of fruit and shoot borer damage. And this has been done, as I told you, for 2 consecutive years in these 3 locations. Now, the next step is to conduct BRL2, that is Biosafety Research Level 2, on 1 hectare, that is 2.5 acres plots. So subsequently, after completing all the biosafety testing, environmental safety testing, and also biosafety research level 1 field testing, biosafety research level 2 testing, the trial has been completed at 1 location in New Delhi. We are yet to conduct the biosafety research level 2 trials on 2 more locations, which are planned for this year, that is 2021. In the month of May, June 2020, the Ministry of Environment and Forests, that has Genetic Engineering Appraisal Committee as part of it, has given permit to Beach Sheetal Company to do BRL2 trials in 5 to 6 different states, that is Maharashtra, Karnataka, UP, Haryana, etc. So we are now waiting for the biosafety research level 2 trials in the Kharif season of 2021. So much so, by the year 2022, the first half of 2022, the data can be submitted, all data, encompassing molecular data, biosafety data, environmental safety data, protein expression data, and the effect of Bt Brinjal on non-target insects data. Non-target insects, mind you, these are all beneficial insects in agricultural ecosystems. So we have to give data about the harmless nature of Bt Brinjal cultivation on, let us say, honeybees, bumblebees, ladybird beetles, mites, spiders, and all other soil flora, fauna, and beneficial organisms, and so on. So these things will be done in the year 2021, and hopefully in 2022, this Bt Brinjal Event 142 will be released to the farmers of India in the next year. I think there I have come to the end of the presentation. I hope I have confined myself to the given time. I have been told 45 minutes is the lecture duration, and whatever time is left, I will be very happy to answer your questions, make any clarifications you want, and feel free to ask any kind of questions. Okay.
Dear all, the session is open for question and answers. If you have any doubts, please ask sir, and sir will explain you the things. So first, I would like to ask one question.
Please.
It's more pertain towards commerce and economics than the science. What could be the fate if the moratorium could be extended further for another 10 years? What could be the fate of the guys and the people who are working on transgenic plants and development of transgenic products, especially the food things?
Yeah, actually, the moratorium imposed by the Minister of Environment and Forests in the year 2010 on Bt Brinjal has a cascading effect on other GE crop testing, development, research, so on and so forth. That is now 11 years. As I told you, February 2010, it has been imposed. Now we are in 2021. February is coming near. It's more than 11 years now. And unfortunately, this particular step that has been taken by the government of India has adversely affected the research in public sector, private sector, the corporate sector companies, the research in universities, research institutes, agricultural universities, and so on and so forth. The transgenic research, unfortunately, has been put on shelf now, and very few people are working on genetically engineered crops. Even the multinational companies that came with a lot of enthusiasm 15, 20 years back, they have stopped working, and some of the companies like Bayer and Dove Agro, they have withdrawn the applications given to Genetic Engineering Appraisal Committee for field trials of maize, rice, and novel cotton events, and so on. So it has very, very, very negatively, adversely affected GE research in India. And we hope that in next 2 to 3 years, there will be some light at the end of the tunnel, and the government of India may hopefully revise its stance on this.
Yes, sir. Guys, if any other doubts, please.
Any other? Please feel free to ask.
Sir, Dr. Anandakumar sir, thank you, sir. I have one doubt. I'm Dr. David. Sir, even like so till your presentation, sir, even mostly the white population knows that some BT, whichever crop named as BT, seems to be somewhat not good for us or something like that. So why can't the press or the government take an initiative to make this is fine and this is good to be used by all? Like why the government is not taking much initiative to be to create awareness to the common people?
Yeah, you are very much right. In fact, the common public has lot of misunderstandings, apprehensions, and wrong notions about genetically engineered crops, especially Bt crops like Bt Brinjal or Bt maize or Bt soybean, and so on and so forth. In fact, the government of India, especially Indian Council of Agricultural Research and Council of Scientific and Industrial Research, has been doing lot of popularization of biotechnology, especially genetic engineering and transgenic crops, through many outreach programs, publishing articles in newspapers, popular science magazines, conducting workshops with farmers in local languages, publishing pamphlets in about a dozen Indian languages. Lot of exercise is being done. But at the same time, the anti-genetic engineering lobbies, anti-biotech lobbies, and there are quite a few NGOs, they are more active and more strident than scientists or the government of India organizations like Department of Biotechnology. So some of the policy makers in the government of India, they are feeling shy of giving approval to Bt crops or even genetically engineered crops. We have, as I mentioned earlier, new events in Bt cotton and also in GE maize, but these have been withdrawn by the companies. And I tell you for your information, about half a dozen transgenic events in important crops like potato, then mustard, maize, have been developed by public research institutes and universities, but they also have been unfortunately put on the shelf. For instance, mustard hybrids developed by Professor Deepak Pental, the former Vice Chancellor of Delhi University and presently working in South Campus Delhi University, has developed genetically engineered mustard hybrids that have 20% more yield than conventional mustard varieties. And this particular GE mustard hybrid has completed all testing, biosafety testing, environmental safety testing, so on and so forth. It was almost approved by the Genetic Engineering Appraisal Committee, but the Ministry of Environment and Forests, as they have done in 2009 with Bt Brinjal, they are very, very reluctant, hesitant to approve Delhi University's GE mustard. So this kind of thing has been happening in the past 7 to 8 years, and it is very, very unfortunate. And as you mentioned, there is a lot of efforts on our part, government part also, to popularize the science of biotechnology, especially transgenic crops or GE crops. But we only hope the government, the policy makers in Government of India one day will listen to us and take a positive decision about this.
Thank you, sir.
Welcome. Any other? Feel free. Dr. Kavikishore, nice to see you. Thank you for joining. Professor Kavikishore, can you hear me?
Sir, please unmute your.
Yeah, I think.
Not about you, sir, Professor sir. Kavkishore, sir.
Yeah, that's okay. That's okay. I think yeah, but I see he's watching.
One more question. Okay.
Yeah, please.
If there is such a strict ban in introducing the BT crops in India, why there are cases reported in Haryana, especially Haryana and parts of Punjab, cultivating Bt Brinjal and mustard? How come they are coming into open market straight away?
My dear, this is not officially approved. You know, as I told you, in the year 2014, the Bangladesh government has approved cultivation of Bt Brinjal. 4 varieties. These are open pollinated varieties. They are not hybrids. So farmers can save the seed and plant it again the next year. And part of that seed came into India through porous border, Indo-Bangladesh border, and it has traveled up to Punjab, Haryana, and it has traveled down to South India, even Andhra Pradesh, Tamil Nadu, and Karnataka. So Bt Brinjal, developed by Mahiko, commercialized by Bangladesh, is now accessible to many farmers in India unofficially. Farmers are growing that. Everybody knows that. Government also knows that. But nobody wants to recognize the truth. Not only that, for your information, the herbicide-tolerant Bt cotton is being cultivated from Punjab, Gujarat, Rajasthan to Telangana and AP. It is available. It is also unapproved. And it belongs to Monsanto or Bayer. Herbicide-tolerant Bt cotton, if you go to fields in Telangana, you see farmers spraying glyphosate herbicide on the weeds. That is because their Bt cotton is herbicide tolerant, unapproved. That is the unfortunate situation in India now. Farmers are cultivating, but government doesn't want to recognize it. Or rather, approve the events that have gone through all regulatory system requirements.
Thank you, sir.
How about students? Any students are listening or not?
They are listening through classrooms, sir. They are listening, sir. More than 288 are listening, sir.
But they don't have any questions, it seems.
Students, please ask questions, whatever you feel.
Feel free.
It's a good chance for you guys to meet
Hello.
some professors. Please ask.
Hello, sir.
Yes,
Speak a bit
please.
louder.
Hello, sir.
Yes, please.
Sir, can I ask question regarding the Bt Brinjal?
Most welcome.
Yeah, sir. Actually, I am from NIPB, National Institute for Plant Biotechnology, Pusa.
Oh, great.
Yeah, sir. Actually, I have just read your papers regarding the Bt Brinjal.
What
and
is your good name?
My name is Zahur, sir. Zahur Ahmed.
Okay, okay.
I am
Please.
working under Dr. Anita Grover, ma'am.
Oh, you are working with that Ranita. Nice.
Yes, sir.
Nice.
Yeah.
Okay.
Sir, actually, I am also working on this one, mustard brassica gentiana. We have
Yeah.
developed transgenic lines.
Yeah, yeah.
And it's one alternaria resistant.
I know.
And also we have just checked the done the fungal bioassay, and it has shown good results outcome. So, but because of that ban in India for the genetically modified crops, so suppose we are doing research in different labs in India. So, and we have developed also some lines in different crops, but it's not approved by the government because of some circumstances like the environment like issues, and some NGOs are just working on it. So we should we should suppose to promote more and more this research in farmers group, as well as in the rural areas, so that they can get just motivated to adopt this crop and raise their voice, and ask the government and demand from the government, instead of suppose motivating this one students and scientists. We should motivate farmers. They are the one who can just adopt that technology.
Yes, it's a very good question. By the way, are you expressing chitinase glucanase in mustard?
Yeah, we have expressed NPR1 as well as chitinase.
Okay, NPR1. Okay,
Yes,
and
sir.
also chitinase. Okay.
Yes, sir.
You see, one should not feel pessimistic just because government of India in the past 1 decade has not been very encouraging. But things will change. As I told you, globally, 29 countries have been cultivating GM crops or GE crops. 44 countries which don't cultivate GE crops, they have been importing
Yes, sir.
the GE crop products like oil and the other products, processed products, they have been consuming. So, and the list is increasing year by year.
Yes, sir.
Year by year. So what
Yes,
will
sir.
happen? Government of India cannot forever close its eyes or
Yes, sir.
close its ears. So at some point of time, all the GE crops developed by us, including NIPB, for example,
sir.
alternaria resistant mustard,
Yes, sir.
may get approval in the near future.
Yes, sir.
So keep your hopes high.
Yes, sir.
Whatever development of a tangible GE mustard resistant to alternaria. And
Yes,
I wish
sir.
you all the best. And
Thank you.
I'm sure GE crops have a golden future.
Thank you, sir. Thank you.
Welcome.
Sir, one more question. Suppose
Welcome. Please.
we have now this one we have genome editing, this technology available.
Yeah,
And
yeah.
recently from Nabi, they have developed this one in banana. They have done the genome editing in banana. And yeah, also they have, I think they are in the process of releasing it.
Yeah, yeah.
Can we do this genome editing CRISPR-Cas in normal cultivated crops?
Yes, yes, yes. In fact, genome editing has become a more powerful tool and technology for improvement of crops.
Yes,
Not
sir.
only in other areas like medicine, human health, pharmaceutical biotechnology, but also in agriculture. Genome editing has a very, very bright future. And many laboratories all over the world have been using genetic editing, genome editing for crop improvement, whether it is in rice or other important crops.
Yes, sir.
And especially for fungal resistance, for nutritional quality, and this kind of important traits,
Yes, sir.
genome editing is very, very beneficial. And
Yes, sir.
also, as you know, the concerns associated with genetic engineering are
Yes, sir.
not by and large present in genome edited crops.
Yes.
That
Yes,
is
sir.
one advantage.
Yes, sir. That's true. Thank you, sir. Thank you.
Welcome.
Thank you for the
Welcome.
explanation.
Any other? Any other?
Any other doubts?
Any students feel free to ask?
Dr. Surendra, do you have any questions? Anybody from students? Students of
Dr. Anil Kumar and Dr. Indira, you please save this PowerPoint and give it to all the students.
It's already live telecast in YouTube also, sir.
Yeah, they can save it in their computers and make use of that in future. Feel free to use it.
Sure, sir. Thank you, sir. Any questions, students or participants?
Okay, as they say, once the presentation is over, and if nobody asks any questions, as they say, everybody understood everything, and there is no need to ask any questions. So I also assume that everybody understood everything. Okay?
Thank you, sir.
Thank you, sir. Thank you very much.
Welcome.
Now, Dr. Charles sir will give you the concluding remarks.
Yeah,
Thank you, sir. Thank you, sir. Sir, I sincerely thank Dr. Anandakumar sir for his wonderful speech on these BT crops. It was really, we had a very interactive, interesting discussion session also. Sir, I would also like to thank Professor Kavikishore sir for organizing this webinar series. And I thank Professor Krupanidhi sir, Head Department of Biotechnology, Vigyan's Foundation for Science, Technology and Research, Guntur, for this opportunity for arranging this webinar series, and we are all good to get together and discuss on these hot topics. I also wish to thank all the fellow faculty members of Department of Biotechnology and the students who have joined in today for this wonderful session. Thank you, sir. Thank you once again.
Thank you. Thank you very
Dr.
much.
Anandakumar, sir. Thank you.
I once again thank Professor Kavikishore Garu for giving me this opportunity to interact with your students and faculty. All the best, sir.
It's a pleasure, sir. Thank you very much for giving us an opportunity.
Welcome, sir.
to
Welcome.
hear to you.
Welcome.
Thank you very much. Now I am closing the session. Thanks for your patience and time and readily accepting our invitation to deliver a wonderful lecture on Bt Brinjal and giving insights into what could be done in the next few years. Thank you, sir. Thank you very much. Thank you.